Good morning, everyone. Welcome to TeleShadowing. We are live on YouTube. As always, feel free to ask questions in the chat box as we go along and your questions will be addressed. So today we're kicking off our Infectious Diseases Month on our Pathway to MedEd series as I'm studying for step one and also reviewing the microbiology section for the step one exam. I'm going to be going over just a couple basics on microbiology. I know personally I took microbiology in undergrad. It wasn't required for medical school, but it did provide me like a very good foundation to understanding all about microbes. Like maybe you've heard of gram-negative, gram-positive. That's also very important. So before we start, I actually wanted to show you all a very interesting video by NPR's Benjamin Arthur and Rob Steen. So basically, I want you to think about like the next time that you look into a mirror, like think about this, like how many ways you can be more microbe than a human, which is very interesting because there are actually 10 times more cells from our microorganisms like bacteria and fungi in and on our bodies than there are actual human cells in and inside of us. So scientists like they are increasingly thinking as more and more research is being published that these microorganisms have a huge influence on our health, and without them our bodies don't seem to do as well either. So there are of course good bacteria on top of the bad bacteria we hear about so often, but there are so many symbiotic relationships in our bodies from bacteria and our microbes. Symbiotic means mutually beneficial to us and to the microorganism living or colonizing inside of our GI tract, for example. So, without the microbe, like we actually don't seem to be as healthy and might actually get sick more often. But these tiny microbes, they're basically invisible, of course, to the naked eye. So, I'm going to show you all basically a visual representation of it all. And I think it's a very good introduction to someone who may not be aware of microbiology before and just how interesting it all is. So I'm going to play the video for everyone now.
The next time you look in the mirror, think about this. In many ways, you're more microbe than human. There are 10 times more cells from microorganisms, bacteria, viruses, fungi, than human cells in and on our bodies. And our genes are outnumbered 100 to 1 by microbial genes. Scientists even have a name for all these microbial genes, the human microbiome. Now, this might make a lot of people rush for the hand sanitizer. But it turns out most of these microorganisms aren't bad, germs that will make us sick. Most are good. And without these good microbes, our bodies don't seem to do as well. We don't seem to be as healthy, and we actually might get sick more often. So one question is, where do our microbiomes come from in the first place? Well, like a lot of things, it starts with our mothers. As the infant passes through the birth canal, it gets coated with microbes from the mom. These microbes may kind of seed the baby with just the right mix. Combined with bacteria in breast milk and other microbes we encounter early on, they seem to slowly take shape in our first few years of life. The overall mix of our microbes becomes very personal, sort of like a fingerprint or maybe a blood type. But our microbes tend to resemble those of our parents and siblings and may stay with us for much of our lives. They may also be doing all sorts of things, such as educating our immune cells, like this one, teaching them the difference between things they should fight off, bad bugs that might make us sick, and things that aren't a threat, like our good microbes. When we're adults, microbes become our first line of defense, fighting off germs that try to invade our bodies, protecting their turf while protecting our health. Scientists have discovered they can even spew out their own antibiotics. The types of microbes in your body vary depending on exactly where they live, like different ecosystems in nature. There are wet places like our mouths, noses, and armpits, oily places like our scalps and backs, and dry places like our forearms. Different species of microbes have adapted to each of these habitats. The biggest, most important microbial habitat seems to be in the gut. It's the most complex, the most diverse, and everything microbes are doing everywhere else in our bodies, fighting off infections, revving up and dampening down our immune systems, signaling cells, that's all happening in the gut in spades. They even seem to help regulate our metabolisms, how much energy we burn and how much fat we store. So if it's not functioning properly for some reason, because of what we eat, antibiotics we take, that may actually lead to all kinds of diseases. Diseases like colon cancer, colitis, maybe even diabetes and obesity. Some scientists think one reason a lot of diseases are increasing is because we've lost key gut microbes. Our microbiomes look far less diverse compared to those of people in less developed countries and earlier generations. And remember how we get our microbiomes in the first place? From our mothers when we're born and from breast milk? Well, some scientists think that too many babies aren't getting that because of all the C-sections and not enough breastfeeding, plus all the antibiotics kids get these days and our obsession with cleanliness. All this may help explain why problems like asthma and allergies have been soaring. Maybe because our microbiomes never taught our immune systems how to work the right way. Maybe swallowing good microbes, probiotics, could prevent and treat some diseases. So could taking prebiotics, essentially food that good microbes love. We end our story with a reminder. This research is really new. We still have a lot to learn about what many of our microbes are really doing. But scientists say that it's getting clearer and clearer that the tiny organisms all over our bodies are essential to our health and happiness.
So I hope that video sort of clarify a lot of things that I'm going to be talking about soon. But this video was published a little over 10 years ago in 2013. So a lot of things have been discovered since then. And so yes, there are actually a lot of documentaries now released like on Netflix, like one called Hack Your Health, Secrets of Your Gut. Really tells you that you really are what you eat, and everyone's microbiomes in their gut tend to be very different, like a fingerprint, like the video said. So I hope that sort of piqued your interest, and also understanding like why are allergies and asthma so much more higher in prevalence in more developed nations like the United States and Canada, Australia, etc. It's actually because of sort of, you can say, this new movement of, making sure everything's very clean and tidy. In fact, like a lot of my infectious disease doctors who taught us the microbiology unit in med school were like, yeah, you should actually like let your kids, now this is with caution, of course, like let them play, let them like eat dirt even when they're like little toddlers and babies. Of course, not all the time, but just that is just to illustrate like how important it is to get, little kids out there in the world, let them explore. And so unfortunately, like the pandemic, for example, prevented a lot of parents from actually taking their kids out to the parks or to the outdoors. And so, we'll see what the ramifications are of that, of course. This is 2024 and that was back in 2020. So we'll see, and just things to really think about and food for thought. So we go on to the next slide. Oh, okay. So I'm going to be basically introducing you all to the world of clinical microbiology, because in undergrad they sort of just teach you the basics of microbiology. Like I remember, and this is if you're not a microbiology major, of course. But I just took one or two classes on microbio, and I just learned like how to plate, how bacteria sort of culture, the different types of bacteria, the different shapes. But I never really learned like the clinical relevance behind it. And so I'm hoping that I can make it all really fun and easy and understandable for you. It might be very overwhelming at first, but remember, this was all like for step one board prep, right? So I'm going to be sort of giving you an easier version of that, and just sort of introducing the topic in a low stakes environment will help a lot. So I'll introduce it, and then we'll talk about like some case studies and some bacterial pathogens. And then we'll talk about some viruses and fungi. And then also we'll talk a little bit about antibiotics and antibiotic stewardship. in terms of how we are becoming more and more resistant to the more antibiotics that are being invented. So I just wanted to get everyone on the same level playing field of what is microbiology. So basically, microbiology is the study of microorganisms, including bacteria, viruses, fungi, and also parasites. Remember that microbes are everywhere. Like if you touch your cheek, you just touched the microbes on your cheek. They're clinically relevant because they're responsible for many different diseases, from like common infections like strep throat to life-threatening conditions like sepsis and pneumonia. Sepsis, another word for that is septic shock. Basically, becoming infected, and then that causes your body to go into shock. Understanding these microbes actually helps us in diagnosing, treating, and even preventing these infections, which is all very important for patient care and also public health. And we'll mostly focus on bacteria today, but we'll also touch a little bit on viruses, fungi, and parasites later throughout the month, and also maybe a little bit like of an intro in this presentation. So, bacteria, they are single-celled prokaryotes who that don't have a nucleus, and they reproduce asexually through binary fission, and also through like transduction, conjugation. Um, we learned a little bit about this for our MCAT prep. If you go over that section, I'm sort of blanking on the page number, but I remember it was in like the MCAT biology books by Kaplan. I remember reading about it. Viruses, they are acellular. They are made of genetic material like DNA or RNA, and they're enclosed in a protein coat. And they actually require a host cell to replicate. So, fun fact, like scientists are still trying to figure out if viruses are quote unquote alive or not, because by themselves, viruses are not able to replicate and be infect or and cause an infection. So, are viruses alive? Question mark. That's still a question, it's still a debate, but that's something very interesting to think about. Fungi, they are eukaryotic organisms. Remember, eukaryotic organisms are multicellular like us, and whereas prokaryotic organisms are single-celled, unicellular. But there can be eukaryotic organisms that are are unicellular, but all prokaryotes are always unicellular. So like fungi, like yeasts, for example, are unicellular, but they're still eukaryotic. And then there's like molds. They are multicellular and they reproduce by budding or by spore formation. And then parasites, they're eukaryotic and they are larger and a little bit more complex. Like they include protozoa and helminths, and they produce both sexually and asexually depending on the species. And I wanted to show you all this cell envelope from the first aid book. A lot of the screenshots that you'll see are from directly from first aid. First aid is basically this book I think I've talked about before that is considered the first aid for when you're studying for USMLE board exams. So this is just step one, so for the first exam. But basically, like bacterial structures, they have many different components with very specific functions. So flagella that are composed of proteins, they're responsible for the bacterial motility. So I'm going to look for yeah, so here's a flagellum here. And these are gram-positive, gram-negative. We'll talk a little bit more in detail in the next slide about that difference. Basically, both of those types of bacteria have flagellae. And then there's pili, and here there's a single pilus, that's pointing at here in the middle of this diagram. A pili or fimbriae, they're basically made out of glycoproteins. They basically help the cell adhere to different surfaces. With the sex pilus, that plays a key role in conjugation. Remember the way that bacteria sort of replicate and divide, and have daughter cells, for example. There are also some bacteria, and it's all very relevant. Like I'm giving you this background because it's relevant to disease. So I remember thinking about this, and I didn't really understand why I was learning. Why is it important to know that bacteria have pili? And why do bacteria have spores? But basically, some bacteria, like certain bacteria types, can also form spores, and spores are basically like a keratin-like coat with the specific types of peptidoglycans and DNA, and they're found only in gram-positive bacteria. And basically, that allows the organism to survive extreme conditions like dehydration, heat, and also exposure to chemicals. So, for example, in the hospital, there's this a bacteria called C. diff, and C. diff is spore forming, which means that it actually doesn't go away through like a hand sanitizer or if you know, wipe surfaces off against the bacteria will stay in that spore. And that spore is sort of, you can say, resistant to like chemicals that are found in like bleach and stuff. So the only way to get rid of them is like through washing or like washing your hands basically. And it's actually one of the leading hospital causing bacterial infections like C. diff. In fact, almost 1/3 of patients in the hospital can have C. diff at any time, but it's not necessarily like an infectious or like damaging or toxic type of C. diff. That's a smaller subset of patients that do have that, but at any time, 1/3 of patients in the hospital can have it because it spreads so fast and it's hard to combat because you know your Clorox wipes aren't going to be very helpful for spore forming bacteria. And that's why it's very important to know which bacteria have spores and which ones don't, because that can tell you exactly how you can get rid of those bacteria or um or have more of them. And then there's another part of the cell called the cell envelope. Um, the cell envelope is another important structure. Basically, there's also the capsule that is composed of polysaccharides, sometimes other proteins, and that helps protect bacteria against phagocytosis or being eaten by some of our immune cells. Then there's also called the S layer or the slime layer. It's not pictured here, but basically that's a loose network of polysaccharides that aid in the surface adherence and also biofilm formation. And that is usually found in like indwelling catheters. And that is why there's a lot of different hospital-caused diseases because we know the catheters are very important for specific treatments or if the patient really needs a catheter, and why it's so important to keep that catheter clean because it can actually cause some infectious diseases if it's not properly um, taken care of. And then there are gram-negative bacteria. Gram-negative bacteria on the right side. Um, they have an outer membrane that consists of an outer leaflet that contain endotoxins, LPS or LOS, and there's also porins, um, porins and other outer membrane proteins or OMPs. And then they also have an inner leaflet made out of phospholipids, seen here. And then there's endotoxins like lipid A that induce something called interleukin 1, IL-1, which is important for the immune responses. And there's also an O antigen, it's not pictured here, but basically there's a lot of things here, right? For example, like, okay, so look, porins that you can see here, they facilitate transport across the outer membrane. There's also something called the periplasm, that is the space between the cytoplasmic membrane and the outer membrane, um, and specifically in gram-negative bacteria. And then in between is basically the peptidoglycan layer in the middle, you can see here. Um, yeah, so basically, this accumulates various different components that exit the cell, like hydrolytic enzymes and also beta lactamases. And these enzymes and beta lactamases are important because it actually helps us recognize what type of microbe it is, based on the specific types. And then there's also a cell wall, of course. We've heard of the cell wall many times. The cell wall of bacteria is made out of peptidoglycan, which is basically this sugar backbone that is cross-linked with peptide side chains by transpeptidase enzymes. And this net-like structure provides the rigid support and protects the bacteria from like osmotic pressure damage. And then finally, like the cytoplasmic membrane, that is a phospholipid bilayer that contains embedded proteins, like we talked about before, like the enzymes. There's also something called penicillin binding proteins. And in gram-positive bacteria on the left hand, there are lipoteichoic acids. They extend from the membrane to the exterior and play a role in inducing TNF alpha and also interleukin 1. And the cytoplasmic membrane is basically the site for oxidative and transport enzymes and is very important for the cell wall to synthesize. So here I'm going to explain to you gram-negative, gram-positive. They're basically a specific type of gram staining. And the more peptidoglycan you have, remember that a sugar backbone, the more you'll stain purple. And so more gram-positive bacterial cells have more peptidoglycan. And so if a bacterial cell stains purple, then that's how we know that it's a gram-positive cell. Whereas in a gram-negative cell, we saw here, there's actually like an outer membrane and inner membrane. Gram-negative cells tend to be a little bit thinner because they have much less peptidoglycan. And if they have less peptidoglycan, they actually stain pink. And that is what we call gram-negative. And a way I remember it is just a personal mnemonic I have is I love the color purple. Purple just makes me happier. And purple is basically just happier as in more, there's just more of it to go around for me. So that's how I know gram positive as a positive color. It's a happy color. I'm happy when I see purple because it is my favorite color. And that's how I know that a thicker cell wall is gram positive. And then gram negative, how I remember that it stains pink is if you watched Mean Girls, and Mean Girls, they have the saying on Wednesdays we wear pink. I know this is very specific mnemonic, but that's sort of how I remember that gram-negative cells stain pink. Pink for the mean girls. Pink mean girls are negative, and they tend to be more skinnier because they'd always like fat shame you, which is that I'm just talking about the movie right now. The how I remember it that they tend to be skinnier, the mean girls, and so that's sort of why their peptidoglycan layers are much thinner. So I hope that made sense as a way to remember it. But basically, like gram staining is a diagnostic tool basically to identify and classify bacteria into the two main groups of gram positive and gram negative based on their various cell wall structures, whether they have more peptidoglycan or less. Um, and yeah, so the gram-negative bacteria actually appears pink due to a counterstain that occurs. And since there isn't a lot of peptidoglycan to resist that counterstain, it will turn pink. Whereas the gram-positive cells, even with the counterstain, they'll remain purple because of the peptidoglycan. So it's very important to know this because the antibiotics that we have developed to treat various bacterial infections, they work differently based on the bacteria's gram-negative or gram-positive status. So for example, like penicillin and other beta-lactams, they're more effective against gram-positive bacteria because they target the thick peptidoglycan layer. Remember, antibiotics, they are made to target bacterial cells. They don't want to target human cells, right? So our human cells don't have this much peptidoglycan, so that's what helps beta-lactam antibiotics work, like penicillin. Penicillin won't actually target our actual human cells unless you have an allergy. And then, like on the other hand, like gram-negative bacteria with their outer membrane, they are resistant to these drugs because like even if you delete the peptidoglycan layer, the cell wall won't like break because the cell wall didn't rely on peptidoglycan to begin with in this gram-negative bacteria. So there are drugs that are made for that too, like antibiotics such as fluoroquinolones, gram-negative bacteria are more susceptible to fluoroquinolones or also aminoglycosides because they target other bacterial processes, like I think RNA synthesis, stuff like that. So understanding this actually helps the doctor guide appropriate treatment decisions because if a patient has a gram-positive disease, you would not want to give them a fluoroquinolone because that wouldn't be as effective. That is why there's something also called broad spectrum antibiotics. And broad spectrum antibiotics, basically, they exist to target both gram-negative and also gram-positive bacteria, but sometimes they have more side effects. So that's where the clinical reasoning and judgment comes in, is making sure that the treatment pros outweigh the cons, right? We want to make sure that you're actually fixing something that is more life-threatening than causing more other life-threatening conditions. And so that's what all that training is for, to understand that and practice that and make sure that we're providing the best possible patient care. So we went over that and like talked about before about gram staining. What I actually didn't know is that there are we already went over this. So um, I didn't know that there were actually other forms of gram staining as well. For example, the Giemsa stain, that's very important for malaria and intercellular organisms like chlamydia. There's also the PAS stain, that highlights glycogen and is used for Ribble disease. The Zeal-Nielsen stain, that actually detects acid-fast bacteria like Mycobacterium tuberculosis, which causes TB. There's also something called the India ink stain, and that identifies like the different capsules of Cryptococcus neoformans, and it's important for diagnosing like cryptococcal meningitis. Then there's also silver stains, and those are used for organisms like Legionella and pneumocystitis. And then there's also fluorescent antibody stains that are very, very specific, and they're used for diseases like syphilis and pneumocystitis pneumonia. And these methods are very important for the rapid diagnosis and also treatment planning. So there's more than just gram-negative and gram-positive that I learned undergrad. There's also many other forms of staining because there's so many other diseases. So scientists have developed different ways to more effectively gram stain them so they're faster. Not gram stain, but actually stain in a different way. So gram positive, gram negative are the name of a different stain, but it's like Giemsa stain. The periodic acids shift stain, the silver stain. And so these are on the right, different mnemonics that first aid has given us to sort of remember it a little bit faster, like PAS, like pass the sugar, because that's glycogen. Glycogen is a type of sugar. That's how I'll sort of remember this specific type of stain. And then there are bacterial virulence factors. Virulence factors are basically used or as tools that bacteria have to establish infections that evade the immune system and damage host tissues. And basically, exotoxins are secreted and can act far from the site of infection, while endotoxins are part of the bacterial structure itself. So it's very important to differentiate between endotoxins and exotoxins, because bacteria can secrete a specific type of toxin. And that is one way that we can tell that this bacteria is toxic to the patient. Like when we were talking about C. diff, it's spore forming. So we want to know, like, while 1/3 of hospital patients may have C. diff at any given time, we actually want to know, is it actually a toxin producing C. diff? Has the C. diff sort of activated in this patient? And so that's how you will be able to tell and quickly diagnose if a patient needs to be treated for C. diff or not. The reason why is because we also want to have antibiotic stewardship, because the more antibiotics that doctors are prescribing, the more that the bacteria will actually rapidly divide and mutate and actually be able to overcome and have increased antibiotic resistance. And we'll talk about that more, I think, later in the lecture. Um, the capsule, basically, we talked about it before, like a specific type of stain to detect capsules. The capsule actually protects bacteria from being eaten by immune cells, and that makes them harder to clear from our immune system. So that's why we might need a little bit more help from antibiotics if they sort of surpass our immune system. And then there are biofilms. Biofilms are protective communities that protect bacteria that form on surfaces. That makes them more resilient and also difficult to treat with antibiotics. And then there's also antigenic variation that allows bacteria to constantly change their appearance to the immune system. And that makes them much harder to detect and eliminate. And I forgot to mention about pili and fimbriae. They basically help bacteria attach to different tissues, which is very important for, like, this is a way that they can actually initiate infections, especially in areas like the urinary tract. Um, so, you can always read this on your own. I know I'm going over this a little bit fast, but remember, this is a low stakes environment. You don't actually have to memorize all this for now. This is just so that you have this at the back of your mind for when you start medical school. But there's a lot that goes into bacterial virulence factors. And of course, the quizzes are all open notes. You can always come back to this slide if there are any questions on it on the quiz. Now we'll talk about bacterial pathogens and also clinical applications, specifically related to it. So we'll talk mainly about gram-positive and gram-negative cells, because I think that is what most people know about before coming into med school. So this is actually an algorithm that is used. I know there's a lot going on, but basically, like, number one, if I know that this bacteria stains either purple or blue, I know that it is gram positive, but then I also want to differentiate it by its different type of shape. So there are there are different, of course, like algorithms. So basically, like, you go through this flow chart and you're able to tell what type of bacteria it is. So let's say I stain the bacteria and I also visualize it under a microscope and it's spherical shape. So that is another word for that is cocci. And so if it's spherical shaped, I'll go down and I'll check, is this catalase positive or catalase negative? Catalase is a specific type of enzyme in bacteria. So like, if it does tell me, if this whatever diagnostic test I do or the tool tells me that yes, this bacteria does come back positive for catalase, like as in it does have catalase, then I will know, then I have to double check like are these circles in clusters or pairs and chains. So if it's also in clusters, that also verifies for me that this is a staphylococcus. And then I want to be able to differentiate further is if it's coagulase positive or coagulase negative. If it's coagulase positive, then that means it's Staph aureus. If it's coagulase negative, then it's sensitive to novobiosin. And if it is novobiosin sensitive, negative, then it would be S. saprophyticus, and if it's positive, then it's Staph epidermidis. So this is just one way to utilize this flow chart to be able to tell exactly how it all works. And if it was catalase negative and it would also and the spheres happen to be in pairs or chains, it'd be a streptococcus. And we'd also be able to tell what type of hemolysis occurs, if it's alpha, beta, or gamma. And then you move on forward, as you go along. So, all the important pathogens in this flowchart, at least, are in bold italics. And there are some other notes down there, but again, you would apply the same thing to the branchy filament shape or the bacilli shape. So this is sort of how the lab algorithm goes in general for when you're identifying different bacteria. But for the exam specifically, what I have to learn is basically like, is this like for example, if it's a group A strep, then and if it's strep pyogenes, I need to remember that it's beta hemolytic. And there are actually different ways it would show up on a specific type of on a specific type of way it shows. So I hope that made a little bit more sense. Now we'll talk specifically about two gram-positive cocci. So again, we're in the cocci world. We're going to talk about two of them. So specifically Staph aureus and Strep pyogenes. So Strep pyogenes is down here. It's beta hemolytic, and Staph aureus is just it is coagulase positive, and it is in clusters and catalase positive as well. So let's talk about Staph aureus. I know this is a lot of information on this slide, but I'm going to summarize it. So Staph aureus is this very versatile pathogen. It's found on the skin and also in the nose of healthy individuals. But it can cause like serious disease when it reaches the skin or enters the bloodstream. It produces like for example, like so Staph aureus is good because it's on our skin and it's very normal. It's in the nose of the healthy individuals, but it can cause, like I said before, very serious disease if it breaches the skin or enters the bloodstream. So remember from the first video that we watched, each microbe that is on our body has adapted to a specific environment. So there are dry environments like our arms, and then there are more wet environments like our mouth, our mucous membranes. So each environment has its own set of microbes. So Staph aureus can actually produce a variety of virulence factors that can help it cause disease once the environment does change of that microbe. So one of the most important ones is called protein A. And if you know what an antibody looks like, it basically binds to the FC or the constant region of antibodies, and that actually prevents opsonization. And opsonization is the way the antibodies sort of work to phagocyte cells or eat foreign bodies of bacteria, and that actually helps Staph aureus evade the immune system because it prevents opsonization and it prevents the normal working operations of our antibodies. Staph aureus also produces coagulase. Remember, we talked about it being coagulase positive, which actually forms clots, and that walls off the infection and protects it from the immune system. Basically, from it's evading our immune system. It's not good for us, but that's how this bacteria will proliferate more. Staph aureus also produces toxins called that causes toxic shock syndrome called TSST1, and also some exfoliative toxins, which can lead to scalded skin syndrome. And a major concern for Staph aureus is MRSA. MRSA, MRSA, stands for methicillin-resistant Staph aureus because methicillin is basically an antibiotic that over time, more and more patients and more strains of this bacteria have had patients become more resistant to it. And so the methicillin-resistant Staph aureus is even worse to have since it's resistant to many common antibiotics, and that makes treatment very difficult. So MRSA infections, they're common in hospitals and also increasingly seen in the community as well. So the clinical takeaway I want you all to have is that, Staph aureus can cause many different like a very wide range of conditions, from like minor skin infections and abscesses to like life-threatening conditions like sepsis, pneumonia, also endocarditis. Um, but this is just all to sort of have in mind. Now, strep pyogenes, also known as group A strep, like Staph aureus, strep pyogenes can also live on the skin or in the throat without causing much harm. But when it invades deeper tissues, it can then become highly pathogenic. So, strep pyogenes is common and is responsible for common infections like strep throat and impetigo. Impetigo is basically having gold-crusted lesions on the skin. But basically, the main common infection is strep throat, and it can also cause neck fash, which is short for necrotizing fasciitis. And in more layman terms, necrotizing fasciitis is what flesh-eating bacteria are called. So one type of flesh-eating bacteria is strep pyogenes, and the more scientific term for it is necrotizing fasciitis. So strep pyogenes, it produces many different virulence factors that enhances its ability to invade tissues and also evade the immune system. There's a key factor called the M protein, and that actually prevents phagocytosis and helps the bacteria stick to the host tissues. And by preventing phagocytosis, you're sort of suppressing the immune system from actually doing what it normally does. In terms of, remember, if you remember what phagocytosis is basically how our immune system will eat and then digest bacteria that don't belong in our bodies. Strep pyogenes can also produce streptolysin O, which can damage host cells and tissues and also contributes to like a more severe infection. There's another capsule called the hyaluronic acid capsule, and that actually helps strep pyogenes evade the immune system because it mimics human tissues, and that prevents the recognition by immune cells. But one very unique feature of strep pyogenes is this its potential for post-infectious complications, like rheumatic fever and also post-streptococcal glomerulonephritis that can actually happen even up to like 2 weeks after infection with strep pyogenes, and they can actually result from like immune reactions to the bacteria, but not actually from the infection itself. So the main clinical takeaway I want you to take from this slide is that like early diagnosis and treatment of strep throat, they're very critical to like preventing these serious sequelae or different domino effect of diseases. And so both Staph aureus and strep pyogenes, they're both very adaptive and very resilient pathogens that can cause many different diseases, and they're both gram-positive, they're both cocci. They're among like one of the most frequent pathogens that you'll encounter during your medical practice. That makes them very important to understand them very well, like the back of your hand. So let's talk about a case study. So I'm going to break down this case study step by step. So I want you to imagine a 10-year-old child who comes to the clinic with a sore throat, fever, and also a red rash. And the parents are concerned because the child has been feeling sick for a couple of days, and now they've noticed a rash spreading over their chest and arms. So first things first, like what do we do? Like you want to take a good look at the symptoms. You want to do the physical exam. You want to take a proper history. So, the sore throat and fever makes us think of a few different possibilities, but one of the most common causes in kids is a strep throat infection, which is caused by strep, streptococcus, pyogenes. So the rash is also very important because like this sandpaper-like rash you can see on the top right corner can also feel rough to the touch, and it typically starts on the chest and also spreads outward, but it actually spares the face. So sometimes during questions like that I see is like, oh, there's rash all over their chest and arms, but there's nothing on their face. So that one key clinical feature that is classic for strep pyogenes. Um, so this type of rash, along with the sore throat and the fever, sometimes there's also a strawberry tongue associated with it, makes us think of a condition called scarlet fever. And scarlet fever is actually a complication of strep throat too, and that's caused by a toxin released by strep pyogenes as well. So next, what we'd want to do is we'd want to basically perform a rapid strep test. This is a quick and easy throat swab that can tell us if the infection is caused by group A strep. Remember, group A strep is just that other name for strep pyogenes. And then in this case, the test came back positive. So how would we diagnose this child? Well, first we'd diagnose them with scarlet fever. And scarlet fever, like we said before, is a result of a strep throat infection caused by Streptococcus pyogenes or Strep pyogenes. So remember, like I'm taking you step by step, like why was it so important to know about like these random toxins that Strep pyogenes causes? Because you wouldn't really understand where the scarlet fever came from in this child if you didn't know the basics and the history. Remember that we talked about before in January 2024 when we talked about the importance of an effective and thorough patient history. Because you can actually, because many clinicians are actually able to diagnose like almost 70% of diseases based on the patient history. So after we got the diagnosis, we did the the strep test. We'd want to treat it, of course. But again, it's very important because like scarlet fever, it's a very serious infection because if it's not treated, it can lead to long-term complications. Long-term complications include rheumatic fever, which can damage the heart, or kidney problems like post-streptococcal, like we talked about glomerulonephritis. Rheumatic fever can damage the heart by damaging the mitral valves. Remember, the mitral valves are in between which two chambers of the heart? Is it on the right side or the left side? Right. So the mitral valves are on the left side of the heart, in between the left atrium and the left ventricle. The tricuspid valve is found in between the right atrium and the right ventricle. So this is why we take the strep throat very seriously, especially when it's combined with a rash. We do need to treat it very like correctly and quickly, of course. And you'd want to treat it with antibiotics. The best options for this patient would be penicillin or amoxicillin, which work very well against strep pyogenes. And remember, strep pyogenes is a gram positive. So for a gram-positive microbe, penicillin or amoxicillin would be your best bet. So, like key takeaways I want you to take from this is like strep throat isn't just a sore throat because it can have complications like scarlet fever. And a positive rapid strep throat test means like the child should get antibiotics to prevent complications like rheumatic fever. And so recognizing the red rash and the sore throat together is very key to diagnosing the scarlet fever. So see how it's very relevant to treating patients and improving their quality of life. Now that we've discussed a very common bacterial infection and how important it is to recognize and treat strep throat promptly to avoid the complications, I want to move on to a different scenario where antibiotics themselves can actually cause significant problems. That is why there's a whole and infectious disease doctors like have a role called like antibiotic stewardship director like at almost every hospital, because antibiotics are sometimes overprescribed. So, like I said before, like antibiotics are crucial for treating infections like the one that we just discussed. But they can sometimes disrupt the balance of normal bacteria in the body, specifically in the gut. And this disruption can lead to an infection caused by different types of bacteria, specifically Clostridium difficile or C. diff, C. difficile. And that's actually known for causing severe bacteria, especially in patients who have recently been on antibiotics. So here's what actually happens. Like when a patient is treated with broad spectrum antibiotics, especially over a long period of time, those antibiotics don't just kill harmful bacteria, they also wipe out the normal healthy bacteria in the gut. And that creates a huge opportunity for C. diff, basically. And C. diff is normally present in small amounts, but then it can take over and cause an infection. So the main issue with C. diff is that it produces two main toxins. Toxin A leads to watery diarrhea and inflammation of the gut, and then there's also toxin B, and that's much more destructive because that actually damages the lining of the colon and also causes a condition called pseudomembranous colitis. And you can actually see in the image on the on the top right here, how the colon looks when it's severely inflamed, and it's covered with those like white patchy lesions. Um, and also like if there's an endomembranous damage, it would be like pale yellowish as well. So usually the people who are at risk for this are those who have are those patients who are in the hospital and they've been on antibiotics recently, like a broad spectrum one like clindamycin or some fluoroquinolones that are also broad spectrum. It's also more likely in people who are elderly, those who have like weakened immune systems, those who are immunocompromised, or also recently have been hospitalized for a long period of time. Um, and let me go back to yeah. So yeah, we talked about the clinical presentation, the risk factors, and yeah. Now let's talk about like very briefly on the treatment of it. First, you want to stop the antibiotic that's triggering this infection. Usually, it's clindamycin that causes the watery diarrhea in toxin A. And then you'd want to treat with like more specific antibiotics that target C. diff, like vancomycin taken orally. Now, in med school, you'll learn that there's IV antibiotics, also oral antibiotics. So in this case, you'd want to give a patient with C. diff oral antibiotic, specifically vancomycin. And then in very severe cases, some patients might need like something called a fecal microbiota transplant. And yeah, like that's actually like a transplant of healthy stool to restore the balance of gut bacteria. And I watched this documentary on Netflix I mentioned before, like The Secret of Your Gut. There are people who actually like take fecal pills from other microbiota. Now, this is not FDA approved yet, but of course a lot of dietary supplements aren't FDA approved. We talked about this before in our nutrition lecture. But I would say it's very interesting because everyone has a different microbiota. So the reasoning here is that you can repopulate your gut from a normal healthy donor with a fecal microbiota transplant. So while antibiotics are life-saving, they can also like disrupt the gut's normal flora and cause serious infections like C. diff. So it is very important to recognize the symptoms and treat this quickly, treat the infection quickly. Otherwise, like all the other healthy bacteria might be surpassed by C. diff and cause some serious complications. So I hope this made sense and this is interactive enough to sort of understand and go through the C. diff and how it's usually clinically presented. Now we'll talk about gram-negative bacteria. So very similar to the gram-positive lab algorithm. This is the gram-negative lab algorithm where when or if or when a gram-negative bacteria stains pink after the counter stain, you want to check under a microscope what shape it is. If it's a diplococci, like as in it's a pair of two spherical shapes or ovalish shapes, or coccobacilli, this is kind of like an ovalish shaped single sphere, or are they curved rods? These are all very important. Or also bacilli, which are just like longer, more cylindrical shaped. So based on that, you want to check if it's aerobic or anaerobic. If it's an aerobic diplococci, and if it ferments maltose, then it would be N. meningitis. If it's a lactose fermenting bacilli gram-negative, then you'd also want to look, is it a fast fermenter or a slow fermenter? If it's a fast fermenter of lactose, then it could likely be E. coli. Um, so again, like the same thing applies if it is oxidase positive, but then you're looking at it in different lab conditions, like different temperatures. It could be like Campylobacter jejuni. So these are all very clinically relevant, especially in the bold and italicized text, because they are very relevant to different clinical diseases and manifestations that you will see in patients with various infectious diseases. Now, there is a whole specialty dedicated to infectious diseases. That is after internal medicine. You have another 2 to 3 years of infectious disease fellowship after internal medicine to specifically focus on the various infectious diseases that can manifest in patients. So moving on, we'll talk a little bit about Enterobacteriaceae. So the Enterobacteriaceae family, that consists of gram-negative rods that are very diverse, that are found in various environments like water, soil, and also the human body. Some of the members of this family are harmless, while others like E. coli, Salmonella, and also Klebsiella, they are major causes of human infections. And these bacteria can lead to conditions like urinary tract infections, like UTIs you may have had in the past, pneumonia, and also gastroenteritis. That makes them highly relevant in both the community and also hospital settings. So let's talk about E. coli. E. coli is always in the news. Sometimes there are foods that are infected with E. coli, or sometimes you're not washing. lettuce properly. I remember recently Costco had to recall a lot of its produce and salads because there were some E. coli infestations in specific branches in the United States. So, E. coli is really one of the most versatile bacteria that you'll encounter. And while most E. coli strains are harmless and actually do live in our gut in a normal, healthy human being, some strains can actually cause infections. So there are three main types that we'll focus on today. So there is ETEC, EHEC, and UPEC. So there's enterotoxigenic E. coli, also known as, well, actually, it's a common cause of traveler's diarrhea. It produces toxins that lead to watery diarrhea. This is the type you often hear about when someone gets sick after traveling to a country with a different water quality. That is why when a lot of people travel to developing nations, they either need to boil their water or drink bottled water if their microbiome is so used to like American or like more developed country cleaner quote unquote water. Um, then there's enterohemorrhagic E. coli, and that's much more serious. It causes bloody diarrhea and actually lead to a dangerous condition called hemolytic uremic syndrome or HUS. That can actually cause kidney failure in children. EHEC is typically contracted through contaminated food like undercooked beef. And then there's also uropathogenic E. coli or UPEC. That is the leading cause of UTIs, and its ability to like basically adhere and stick to the lining of the urinary tract. So remember I was talking about like pili and fimbriae. Like why are those specific parts of the bacteria so important to remember? Is because in uropathogenic E. coli, they have this really good ability to adhere to the lining of the urinary tract with special structures called adhesins, and that makes it very effective at causing infection. So these different strains highlight like how versatile the E. coli bacteria is, often causing like anything from like mild diarrhea to life-threatening conditions like HUS. So it's very important to understand like the specific strains and how to operate to diagnose and also treat the E. coli infection very effectively. But yeah, so these are different valence factors that are associated with E. coli as well. You can read through at your own leisure. Now this is again, I talked about this already, the different types of E. coli and how it's so versatile. And now we'll talk about a little bit on a case study. So here we have a 4-year-old child who has been brought to the ER by their parents due to bloody diarrhea and also abdominal pain that started 2 days ago. The child has no fever, but the parents are concerned, understandably, because the diarrhea has become bloody and the child hasn't been eating or drinking much. So, upon further questioning, the parents mentioned that the child had an undercooked hamburger at a family barbecue about 3 days ago. This timing fits the typical incubation period for certain foodborne bacterial infections, which actually makes us think that the food might have been the source of the infection. So, after getting the history, you do the physical exam. And so, on physical exam, the child is mildly tender in the abdomen, but is otherwise showing like not many to like to not no signs of dehydration or any severe distress. And so this is important because severe dehydration can be a major complication of gastroenteritis, especially in children. So that's how you can actually narrow down your differential diagnosis. So we've done a preliminary stool test, which has come back positive for Shiga toxin. Now, if you remember on the previous slide, we had Shiga toxin on there. But we're still waiting on the complete stool culture results. But the presence of the Shiga toxin, that is a big clue, because this toxin is produced by EHEC, the enterohemorrhagic E. coli, one of the dangerous strains of E. coli that we discussed earlier. So again, this is how understanding the specific type of bacteria can help you diagnose something much faster when you're still waiting on lab results, because sometimes you need to treat the patient faster then the labs can come back results with. Oftentimes, labs should be more confirmatory rather than exploratory for your differential diagnosis. Um, and here now we'll talk about viruses and also some fungi. Now that we've talked a bit about bacteria, now I'll talk briefly about these, but we'll talk about it in more depth throughout the month. So I will be very brief and we'll touch on a few things, but we need to really delve deeper into viruses and fungi as well. So unlike bacteria, viruses are much smaller and much more simpler, but they can cause just as much or if not more damage. So, like, what exactly is the virus? If you'll remember from your MCAT prep days or in your future MCAT prep days, viruses are basically just packets of genetic material. It can be like either DNA or RNA, and they tend to be wrapped up in a protein coat called a capsid. And some viruses also have an extra layer called an envelope, and that actually, and like basically the envelope is what they steal from the host cell's membrane as they leave the cell. And these structures are important because the capsid protects the virus's genetic material and the envelope when it's present, and that can actually help the virus infect new cells. So I hope in the back of your mind you're thinking, okay, then that means that antivirals or any treatment that's targeted towards viruses should target the capsid. Because if the capsid hasn't targeted of a virus, then that means that the virus won't be able to enclose its genetic material properly. Like just like the nucleus of a cell, if our genetic material is not properly enclosed in the DNA, in that, that double helix bound DNA, it does not function as it should. So the reasoning behind what I'm saying is when I'm talking about capsid, things that are unique to viruses only, is how you should be thinking about, okay, maybe this treatment should target the viral capsid because our eukaryotic, like normal animal cells in our bodies, like they don't have capsids. So that is one way to sort of hack into viruses only in any treatment that we may have. So, and one thing to also remember about viruses is that they cannot replicate on their own. So they need to enter a host cell and hijack its machinery in order to make copies of themselves. So this is actually what makes viruses so tricky to deal with because to kill a virus, we usually have to target it inside our own cells. So let's break it down into two different types of viruses. So there are DNA and RNA viruses. And DNA viruses, they tend to replicate in the nucleus of the host cell, and they're usually a little bit more stable when it comes to mutations. And on the other hand, there are RNA viruses, and they typically replicate in the cytoplasm. And they're known for mutating much more rapidly, and that's why viruses like the flu and HIV, which are both RNA viruses, they can evolve quickly and evade the immune system. So, something I want you to take away from this slide is that viruses are basically like genetic material wrapped up in a protective coat, and they rely on a host cell for replication. So, knowing whether a virus is DNA or RNA based actually helps us understand how it replicates and how we might target it with treatments. So, here is a much more simple comparison between DNA and RNA viruses. So, DNA viruses, like I said before, they replicate in the nucleus and they have a much lower mutation rate because the DNA polymerase enzyme has this built-in proofreading function like we talked about during our not microbiology, our cellular biology lectures in the past. And that actually makes them much more genetically stable. So an example of a DNA virus includes the herpes virus and also hepatitis B. And on the other hand, the RNA viruses that use RNA as their genetic material, they replicate in the cytoplasm, like we said before, but they have a much higher mutation rate because RNA polymerases don't have that proofreading ability that DNA polymerases have. And so that's why viruses like the flu, aka influenza, and that's why you have to get our flu shots renewed every year, and also HIV, they mutate so quickly, which allows them to evade the immune system and also require frequent updates in vaccines. So DNA viruses, they are much more stable, but they replicate much more slowly. And RNA viruses mutate much more quicker, and they make them much more unpredictable and also harder to control. So, understanding these differences helps us approach treatments and vaccines more effectively for all of you future doctor scientists out there. Now, let's talk about influenza or the flu, which is one of the most common RNA viruses that causes significant illness every year, especially during the flu season. So, influenza virus has two key proteins that help it infect us. So, there is the protein, aka hemagglutinin, and it helps the virus attach to our cells and also get inside of them. There's also the neuraminidase NA protein that acts like scissors that basically cuts the virus free from the host cells so that it can spread to new cells. And these proteins are also the targets of flu vaccines and also antiviral medications. You've probably heard of medications like Tamiflu that actually works by blocking the neuraminidase that actually stops the virus from spreading. And clinically, as you may know, I know I've had the flu myself many times. The flu usually causes fever, cough, muscle aches, and fatigue, but it can be much worse for vulnerable groups like the elderly, young children, or people with weakened immune systems. Because one of the major causes of concern is pneumonia, which can be either caused directly by the virus or as a secondary bacterial infection, often through Staph aureus. Remember, I talked about how Staph aureus tends to be in the nares, in the nose, in our nostrils. So but I talked about how if Staph aureus breaks into our skin barrier, remember, like when you have such a intense flu, sometimes, I know sometimes it can sometimes lead to nosebleeds if you're blowing your nose really hard. But again, that is one way Staph aureus can actually enter your bloodstream. But the best way to prevent the flu is through the annual flu vaccine, and that helps your immune system recognize the HA and NA proteins on the virus. So a key takeaway that I want you to take from this slide in influenza is that, of course, the flu is very highly contagious. It mutates frequently, similar to COVID-19, and that is why we need a new flu vaccine every year. So understanding how the virus enters and leaves cells is crucial for both the vaccine development and also antiviral treatments. So I hope this serves as a reminder for you to get your flu vaccine. I got my flu shot earlier this week as well. So it is flu season. It is September 2024. So definitely get your flu shot in a timely manner. So here's a case study I wanted to talk to you all about. Here we have a 65-year-old male who presents with fever, cough, body aches, and fatigue for the past 3 days. He didn't get his flu shot this year and recently attended a crowded indoor event, which increases his risk for exposure to the flu virus. After getting his history, he wanted to do a physical exam. On physical examination, he has mild crackles in the lungs, which is something you hear when you have your stethoscope and you're listening to the lungs and lung sound. That could actually indicate early signs of a respiratory infection. His temperature is also elevated at 38.5 degrees Celsius, and 38.5 degrees Celsius is approximately 101.3 degrees Fahrenheit. So a 101 degree Fahrenheit fever. That would actually fit his flu symptoms. Um, and then we actually performed a rapid flu test, and the result came back positive for influenza A. Now, there's also another influenza type called influenza type B, but this one is influenza A. And given his age, this patient is at a higher risk for complications. I guess this is a 65-year-old male. So, what is the likely diagnosis, and what complications should you be concerned about in this patient? This is a question for you all in the chat box. So, what is the most likely diagnosis? We just talked about it in the previous slide. And what should we be most concerned about in terms of complications? Okay, good. I see flu. So you're right, influenza is what we're concerned about for the diagnosis, and especially after the test said it is positive for influenza A. What complications should we be concerned about? Yes, pneumonia, and also like a secondary bacterial infection from Staph aureus. So you're right. So this influenza was confirmed by the positive rapid flu test, and while most people can recover from the flu, there are elderly patients like this one who are at a higher risk for complications like pneumonia, either like a viral or also a secondary bacterial infection. So we should definitely closely monitor this patient for any signs of worsening respiratory system, for any worsening respiratory symptoms like increasing crackled in the lungs or a difficulty breathing, which could suggest pneumonia. So treatment will likely involve supportive care measures at this time, and in some cases we can use antiviral medications like Tamiflu to reduce the severity of the illness. So influenza overall, like all in all, can be serious, especially in older adults. And vaccination is key to prevention. Now, getting the flu vaccine does not necessarily always mean you won't get the flu. It just means that you'll have reduced symptoms because your body has already been exposed to it through the vaccine. And then rapid testing can actually help guide your treatment early on. Now I'll give a very brief overview of fungal pathogens. So they are a bit different from bacteria and viruses. Like fungi can come in two main forms, aka yeasts and molds. And some fungi can switch between these forms depending on the temperature, which makes them very dimorphic because they can switch between being a yeast or a mold. Yeasts like Candida albicans, they are unicellular and they can cause infections ranging from thrush in the mouth to vaginal yeast infections to invasive bloodstream infections in hospitalized patients. So remember that video, that introductory video I showed you in the beginning, that when a baby is born through the vaginal canal and the baby is being born through that way, and if the mother has Candida albicans, this is how the baby can have it. So this is why it's important to remember, like, because there are microbes everywhere, on us, in us, everywhere. This is how, like, sometimes people are scratching their heads, like, how did this newborn child get this, like, Candida albicans? Might have probably been through the mother. So besides that, there are also molds. Like the molds are multicellular and they form long filaments called hyphae. And there's a species called Aspergillus. They can cause respiratory infections, especially in people who have like weakened immune systems, like those with cancer or people with HIV or AIDS. And fungal infections are basically a very major concern for immunocompromised patients, meaning people whose immune systems aren't working as well. And this could be due to chemotherapy, HIV, or even like certain medications like steroids, or of course, immunosuppressants. Um, Most fungi spread through spores, which are often inhaled from the environment, and that actually makes respiratory infections the most common type of fungal disease because you can breathe in those spores, and those spores, if they can proliferate in the lungs. And so fungal infections, what I want you to take away from this slide, like they are usually opportunistic. Opportunistic meaning they affect people with weakened immune systems. So they are taking the opportunity of this person with a weakened immune system and infecting them. Um, and they're also often harder to treat than bacterial infections because fungal cells are more similar to human cells because they're eukaryotic, and that makes drug targeting a little bit more complicated. Now let's talk about the common yeast, Candida albicans. This is a yeast that normally lives on your skin and inside your body, particularly in the mouth, gut, and the vagina. It's part of our normal flora, of course, but when the balance of bacteria and fungi is disrupted, it can overgrow and cause infections. So in its yeast form, Candida is harmless and a normal part of our microbiota, but when it shifts into its pseudo-hyphae form, that is when it becomes pathogenic and can invade tissues. Some common diseases caused by Candida include thrush, which is an oral infection that leaves white patches in the mouth and is common in infants and also immunocompromised patients. There's also vaginal candidiasis, or a yeast infection. Is often seen in women, especially after antibiotic use, which disrupts the normal flora. There's also invasive candidiasis, and that occurs when Candida enters the bloodstream, usually in the hospitalized patients who have weakened immune systems. And this is a serious infection, especially in the ICU patients, because they have catheters in and also IV lines. So the main risk factors for Candida infections are anything that weakens the immune system, like recent antibiotics, hospitalization, or immunosuppression. And treatment usually involves antifungal medications like fluconazole, and in more invasive cases, there can be stronger antifungals like amphotericin B that might be needed. So remember, like Candida is an important part of the body's flora, but it can cause opportunistic infections when the body's defenses are down, which make it a major concern in the hospital setting and also like post-hospital settings as well. And again, this is actually like albica refers to white. This is another reason, like knowing sometimes like the prefixes or suffixes or Latin terminologies can be helpful. And exactly what I said is summarized in this first aid screenshot I took, the common yeast. Now let's talk about resistance and antimicrobials. So, antimicrobials, which include antibiotics, antivirals, antifungals, antiparasitics, they're important tools in modern medicine, and they come in many different forms that target each of the different types of pathogens. So antibiotics, they target bacteria, antivirals, they work against viruses, antifungals, of course, target fungi, and then antiparasitics are used to target parasites. So each of these agents, they work in a different way. So for example, penicillin, which is an antibiotic, it targets cell wall synthesis. That makes it very effective against gram-positive bacteria like Staph aureus. Other antibiotics like tetracyclines, they actually inhibit protein synthesis. While fluoroquinolones, they disrupt nucleic acid synthesis. Remember, nucleic acids are the building blocks of DNA and RNA. Well, RNA is ribonucleic acid and DNA is nucleic acid, but you get the idea. So understanding how these drugs work actually helps us choose the right one at the right time for the specific person, for the right person, for the right condition or specific condition or infection, and also avoid unnecessary or ineffective treatments. So the different antimicrobials, they will all target different specific types of pathogens. That's why pharmacology is so vast and expensive, because you want to have the right medication, the right time for the right individual, and that is called precision medicine. And that's why it's important to choose the right drug for the right infection. And when we use antibiotics, it's crucial to understand how they work, right? Because understanding the different antibiotic targets for different because they target different bacterial processes. I alluded to this earlier in the lecture when I was talking a little bit about like the bacteria structures and why it's important to know. So remember, I emphasized the cell wall. Whether you have more peptidoglycan, that's gram positive, that is purple staining, so gram positive, or you have less peptidoglycan or your thinner cell wall as a result, aka you're gram negative, aka the color you stain is pink. So there are cell wall synthesis inhibitors that include penicillin and cephalosporins. They target the bacterial cell wall, specifically the peptidoglycan layer, and that weakens the cell wall, causing the bacteria to burst. And this is very effective against gram-positive bacteria like Staphylococcus and also Streptococcus. There's also protein synthesis inhibitors like tetracyclines, macrolides like azithromycin. They stop bacteria from making proteins by binding to their ribosomes. Remember, ribosomes are where proteins are synthesized in our eukaryotic cells as well, but these are inhibitors of protein synthesis of prokaryotic ribosomes. And so that's why it's very important to know the difference between prokaryotic ribosomes and eukaryotic ribosomes. And if you'll remember, there's 50S and 30S, which makes 80S, and then I think there's like the 70S one for the 40 and 20, something like that. But basically, like the eukaryotic ribosomes are bigger than the prokaryotic ribosomes, and that's actually how the protein synthesis inhibitors like tetracyclines will not affect our ribosomes. They will affect the bacteria's ribosomes. And yeah, like without protein synthesis, the bacteria can't grow or reproduce. And then there are nucleic acid synthesis inhibitors like fluoroquinolones and rifampin. They block DNA replication or RNA transcription. Remember the central dogma of the cell? Well, bacterial cells also have that. So this prevents the bacteria from reproducing, that ultimately leads to their death, of course. And so each antibiotic class like has a specific site of action. So choosing the right one depends on the type of infection and the bacteria involved. So I believe I misspoke. It's actually 50S and 30S make up the prokaryotic ribosome, and it's actually the 20S and 40S that make up the I can't actually let me look it up right now. I was teaching this to a student earlier. Eukaryotic ribosome. Sorry, there's just so much to remember sometimes. Oh yeah, so basically S stands for small subunit. So the eukaryotic ribosomes have a 60S and 40S subunit. And assembled together, it's called the 80S. And then the prokaryotic ribosome has 50, 30, and 50 and 30S. So how I like to remember it is a little bit simpler. So how I remember is 5 and 3, they're both prime numbers. Prime numbers are like prokaryotes, and also they're lesser, right? 50S and 30S. Whereas eukaryotes are even, E for eukaryote, and also E for, us. I consider us to be more even. And so since we're bigger and multicellular, we have 40S and 60S. And 4 and 6 are both even numbers. And that's how I remember that the eukaryotic ribosome has 40S and 60S small subunit equaling 80S. They both equal 80S, but in different ways. So each antibiotic class has their own site of action, a specific site of action. So choosing the right one depends on the type of infection and also the bacteria involved. So I hope that I built upon this foundation, starting off with what is a microbe, where do they exist, and also the different types of microbes and their specific parts, because understanding all that will eventually lead to understanding diseases, their complications, and then eventually their treatments, because pharmacology is all based on the specific components of the bacterial cell. And if you don't have this foundation of understanding what is this specific part of the bacteria that this drug is affecting, you won't really understand how the drug works because patients will ask you, how does this drug work once you prescribe them something? And so you need to have all these answers, of course, when you're treating and working with human lives. But one of the biggest challenges in using antibiotics is resistance. Bacteria have evolved actually, like I said before, many different clever ways to actually evade the effects of antibiotics, making some infections much harder to treat. So we can actually look at the more common mechanisms of resistance as well. So the first one is efflux pumps, and they are like tiny pumps in the bacterial membrane to actively remove antibiotic from the cell. And that's very clever. It prevents the antibiotic from reaching its target. Then there's beta lactamase production in many bacteria like MRSA or the methicillin-resistant Staph aureus or MRSA MRSA. It produces enzymes that break down beta-lactam antibiotics like penicillin, and this is one of the most common resistance mechanisms. Another way is through altered targets. So some bacteria actually mutate the proteins that antibiotics typically target. For example, in MRSA, it actually alters its penicillin binding proteins. So antibiotics like methicillin cannot bind to them as effectively. So antibiotic resistance is a growing problem worldwide, which is why it's so important to use antibiotics carefully and only when necessary. So here's one of our final case presentations. Here's a case of a 45-year-old male who recently underwent surgery and now presents with redness, swelling, pain, and also purulent discharge at his surgical incision site. You can see here, yeah, in the red area at the bottom. These are all signs of a post-operative wound infection. His temperature is 38.3 degrees Celsius, also known as 100.9 degrees Fahrenheit, which is indicative of a mild fever. And we collected a wound culture, and the results came back showing methicillin-resistant Staphylococcus aureus, or MRSA. This is an important diagnosis because MRSA is resistant to many of the common antibiotics, like methicillin, of course, is in the name, that we would typically use for staph infections. So again, another allocation. And here are antifungals and their targets, but for time purposes, we'll go over this actually, I can go over it briefly and we'll talk about it in more detail when we talk about fungi. So, basically, antifungal medications treat fungal infections, especially in immunocompromised patients. Remember that fungi are also eukaryotic organisms like human cells, so it's much more difficult, or a little bit more difficult, to find drugs that can target fungi without also harming our own cells. But we do have a few classes of antifungals that exist, like polyamines. They bind to ergosterol, which is a key component of fungal cell membranes that we don't have, and they create holes that cause the fungal cell to basically disintegrate and die. And the most well-known drug in this class is amphotericin B, but it comes with serious side effects, especially kidney toxicity. There's also azoles. These drugs like fluconazole, they inhibit ergosterol synthesis. I know there's fluconazole shampoos actually that help with reducing dandruff in the scalp. They actually weaken the fungal cell membrane, and azoles are commonly used to treat infections like vaginal yeast infections and also thrush. Then there's echinocandins, and these are the newest class, and they work by inhibiting cell wall synthesis in fungi. And caspofungin is actually an example of an echinocandin, and it's especially useful for treating invasive candidiasis or also aspergillosis. So each of these drugs has specific uses depending on the type of fungal infection and the patient's condition. So we need to choose them carefully based on the patient's infection and also their health status. And with all that said, I'm going to wrap up today's cell shadowing session. And the link to the TeleShadowing quiz is now live. So, and as always, we'll need a 70% or higher to pass and receive certification. And we look forward to seeing you at future TeleShadowing sessions as well. Thank you so much, everyone, and looking forward to seeing you at next week's sessions. Take care. Bye bye.